Thanks, John. Thanks for the invitation to speak today. So I'm going to say, obviously, a little bit more about mitochondria and the disease and genetics. And as Mike has already commented on, the way we understand mitochondria is that they are, to make a mitochondrion, you need about 1,200 or so genes from the nucleus encoding mitochondrial components and the 37 from the mitochondrial DNA. So I'm going to go give you a quick overview of mitochondria and disease, prevalence, a very quick overview of mitochondrial DNA genetics, say a little bit about treatment, and unfortunately there's not very much to say about treatment really, emphasising the need for prevention. So to start off, I'll mention one condition, Lee syndrome, which is the most common single disorder of mitochondrial dysfunction that we see in children with mitochondrial disease. So in this condition, children are typically healthy for the first 6 months or so of life, then usually after a viral illness undergo an episode of an encephalopathic episode, which they may recover from. That may involve an altered respiratory rhythm, loss of consciousness, funny eye movements, all sorts of things. Typically they'll recover from that, but the standard course is having another episode and a progressive decline, usually accompanied by a build-up of lactic acid in the blood, and in Lee syndrome typically dying by about 3 years of age, although there's wide variability there. The classic features of Lee syndrome are, on imaging, you see these symmetrical lesions in the basal ganglia or midbrain, some of the ancient regions of the brain. And so that sounds like a fairly well circumscribed condition. So what do we know about the evolution of Lee syndromes? Is a bit small to see, but just to remind me to really say that, so Lee syndrome was described from the neuroanatomical picture in 1951. In the 1970s, there were some defects in mitochondrial energy generation recognised at the biochemical level, so finding enzyme defects associated with Lee syndrome. And then the first genetic cause in 1991, so 40 years after this clinical condition was identified, showing that mutations in a mitochondrial DNA gene could cause Lee syndrome. And then subsequently, mutations in the mitochondrial DNA, autosomal genes, or the X chromosome associated with Lee syndrome. So this one apparently fairly well described condition actually appears to comprise at least 75 single gene disorders. And so, the clinical diagnosis of Lee syndrome means nothing really about the inheritance of this condition, for example. And that's one mitochondrial condition with very specific lesions in certain brain areas, but you'll find in other mitochondrial disorders that almost any area of the brain can be involved. Widespread cortical degeneration, leukodystrophy, specific cerebellar abnormalities, and so on. So mitochondrial disorders, you can have many different mitochondrial disorders causing the same pathology in very isolated regions of the brain or in specific tissues, and you can have almost any pathology caused by different mitochondrial disorders. So, the classic sort of description, originally I think coined by Arnold Munich and Pierre Austin, was almost any symptom with any age of onset affecting any organ system can be caused by mitochondrial energy generation problems, particularly those organs that have a high energy requirement, like the brain. Mike mentioned that we are estimated to turn over something like 65 kilograms of ATP every day, and about 20% of that is in the brain and maybe 10% in the heart, and I think about 10% in the kidney and so on. And so there are certain organ systems that have very high energy demands, but almost any organ system can be involved. Early death is very common in these disorders, but there's a wide range in severity. Often there's a progression in severity, sometimes an increasing number of organ systems involved. One of the key problems has been that historically there's been no gold standard diagnostic test, meaning that many families have suffered a diagnostic odyssey of months, years, decades to get to a diagnosis. That is finally starting to turn around with the advent of these new next generation DNA sequencing techniques, which make it much easier. So you can imagine just with Lee syndrome, before we'd be sequencing one gene at a time, and a third of the genes haven't been described until about 3 years ago. So it could take a very long time to get a diagnosis. Now we can sequence all of those genes at once. Unfortunately, we don't have a funding mechanism that really allows us to do that in a diagnostic context yet in Australia, but it's now possible, and there are major efforts funded by the state government and about to be funded by the federal government, $25 million each, that are going to improve that and help the introduction into routine practice. So how common are these disorders? Just to give you an idea of where the numbers come from, I think still probably the biggest and best study of paediatric disorders is one that we published in 2003, looking at a 10-year birth cohort in southeastern Australia, where we looked at basically just the number of births in that period and the number of patients diagnosed with definite mitochondrial disease, and came up with this figure of 6.2 per 100,000 births. We've identified another 20 or so patients from that period since then who developed symptoms in childhood, so we can now say it's a little higher. Perhaps to our surprise, the figure was 12-fold higher in the New South Wales Lebanese community, and that simply reflects that at the time particularly it was the largest group in Australia where there was a lot of consanguinity. So, for example, cousin-cousin marriages, and many of these disorders are autosomal recessive. There are so many different genes involved that any group where there's a lot of consanguinity means that your partner is likely to be carrying perhaps a mutation in the same gene that you are, more likely than the general outbred population, and so any consanguineous group would expect to see lots of these disorders. So in adults, the best data have consistently come from Newcastle upon Tyne, and the latest figure that's published was 12.5 per 100,000. That's for a point prevalence in the population of adult onset disease. You can essentially add those two numbers together to get a minimum estimate of how many children will develop mitochondrial disease during their lifetime, and it's about 1 in 5,000 births, the figure that Mike quoted before. So it's roughly half in adults and half in children, and roughly it's about half mitochondrial DNA mutations and half nuclear DNA mutations. So 1 in 10,000 people are diagnosed with a mitochondrial DNA mutation causing disease, but again, as was mentioned, it looks like 1 in 200 people are carrying these mutations. So there's a bit of a disconnect there. Some of the time that's because many of these people only carry a very small amount of the mutation. They'll never suffer symptoms, but in an unknown proportion there are patients who are suffering symptoms from those mutations that have not been investigated. Again, I think I won't spend time on this because Mike's already done so, but yeah, our current estimate, we keep updating the catalogue. There's another 20 genes or so discovered each year, so our current count is that maybe 215 genes have been identified as a cause of mitochondrial disease, and that probably accounts for 50 to 60% of patients diagnosed with mitochondrial disease. So I won't spend any more time on that. So in the Australian context, we've acted as the major paediatric referral centre for 2 to 3 decades, I guess, and so what do those numbers end up looking like? So we've diagnosed about 600 children with mitochondrial OXPHOS disorders, and our most recent count was that 128 of those children had mitochondrial DNA mutations in a range of different genes. Don't want to dwell on the details too much. Initially, we had very few patients diagnosed with mutations in nuclear genes because there were just so many. Now we're up to about 170, and actually we've just done a new study, and there's another 30 or so diagnoses there already. And to emphasise the difficulty of doing this with traditional technology, so those 170 children, that's finding mutations in 44 different genes, 17 of which we, and I use the royal we a lot because we meaning my group and our collaborators in Sydney and Boston and elsewhere in Melbourne, have identified 17 of those 44 genes as being disease genes, so a lot of work going into finding those diagnoses. And also we've skipped off the bottom there, but there's only 3 of them where there's 10 or more patients with mutations in the same gene. So there's not a lot of common genes to go looking for in nuclear disease. All of the ones that have 10, 26, 51 are basically because there are ancient European founder mutations in the population that come together even in an outbred population. So those particular disorders are more common. So that's a very quick overview of the genetics of the genetic basis of mitochondrial diseases. In terms of treatment, the one-liner, I guess, is that with colleagues in Newcastle upon Tyne and in Finland, we updated our Cochrane review in 2012, which had the very sombre conclusion that after reviewing about 1,000 publications, that at least at the Cochrane standard of evidence, which is really double-blind, crossover, randomised controlled trials, there's currently no clear evidence supporting the use of any intervention in mitochondrial disorders. That's partly a statement that this is a group of rare disorders and it's very hard to do properly controlled trials to get evidence at that level, and it's an overstatement because there's no doubt that we can effectively treat some symptoms, at least for some period of time. Aggressive management of infections is important. Diet, good high-quality diet's important. Mitochondrial cocktails of vitamins can be beneficial in specific conditions like MELAS. Arginine appears to be very beneficial. So, it's just that there's not the RCT level evidence that things are effective. there is, I think, a lot of encouragement coming from studies as we've started to understand the genetic basis of mitochondrial diseases. There's been research in a lot of areas of approaches to improve mitochondrial function, and many of these seem very promising, at least in animal models and some preliminary human trials. Things like trying to prevent reactive oxygen species causing damage, boosting the numbers of mitochondria. When you can't oxidize NADH at complex 1, you have a redox imbalance in the cell, and trying to address that by boosting the levels of NAD, by trying to improve mitochondrial quality control, or by so-called bypass therapies and mitochondrial gene therapies, where they're using gene editing approaches to try to cut the mutant mitochondrial DNA and leave healthy mitochondrial DNA there. So there's a lot of promise there, but most of these are still really some way from the clinic, particularly as we get down the bottom of that list. So I guess that really emphasises the fact that we don't have effective treatments. We at least want to be able to offer families reliable means of preventing mitochondrial disorders. And, two categories here. For nuclear encoded disorders, they're really like any other types of genetic diseases. If you've identified mutations in a gene, then you can offer families options like donor gametes, so typically donor sperm IVF, or prenatal diagnosis, or pre-implantation genetic diagnosis. For the half of the mitochondrial diseases caused by nuclear disorders, we have options that can be made available to families. The complicated ones are mitochondrial DNA, where the genetics is really rather different. So again, partly to give you an idea of what's happening now, and I know Mary is going to cover some of the same ground, but then extend to the mitochondrial transfer type approaches. I thought I'd give you the quick primer on the unique quirky features of mitochondrial DNA that are relevant to offering reproductive options. And I guess the major ones are that mitochondrial DNA is inherited maternally, and that is largely about weight of numbers. So the sperm contains about 50 mitochondrial DNAs. They do enter the egg at fertilisation, but they're grossly outnumbered by the 200,000 copies of mitochondrial DNA in the mature human oocyte. So half the total DNA in the oocyte is mitochondrial DNA. And so the paternal mitochondrial DNA is grossly outnumbered, and then the sperm are targeted for destruction as well. So we see maternal inheritance. There's been one exception to the rule of paternal mitochondrial DNA disease, but in medical genetic terms, mitochondrial DNA is maternally inherited. And then the other features that are unique are heteroplasmy. So if you have a mutation in mitochondrial DNA, it's not like a nuclear gene where you've got 0, 1 or 2 copies of the mutant allele. If you've got thousands of mitochondrial DNAs in a cell, then you might have no mutant DNA, 1% mutant DNA, 5%, 90%, and so on. So you have a heterogeneous cytoplasm. And in these two phenomena, the threshold effect and the mitochondrial DNA bottleneck, that are relevant to options. And I think they're probably best demonstrated by showing a pedigree here, where we look at a particular mitochondrial DNA mutation in this family at nucleotide 8993, causing a syndrome known as NARP, neuropathy, ataxia, retinitis pigmentosa. And so the affected individual in this family has about 80% of their mitochondrial DNA in a range of tissues carries this particular mutation, and about 20% is wild type or healthy mitochondrial DNA. So that patient is heteroplasmic, mixture of mutant and wild type DNAs, and he has 3 sisters. 2 of them have very low amounts of the mutation. One has quite a high amount, 60% or thereabouts, but is healthy. And the reason she's healthy and the boy's affected is that for this mutation, if you have less than about 60% mutant DNA, for this mutation, which is probably the best behaved mitochondrial DNA mutation, the easiest to make these sort of inferences about, if you have less than about 60% mutant mitochondrial DNA, it's extremely unlikely you'll develop symptoms, and then the more mutant you have, the more severe the symptoms are likely to be. So that's a threshold effect. Below that level, no symptoms. Above a certain level, symptoms. And then the bottleneck is that, if you thought that in every cell we had thousands of mitochondrial DNAs, when that cell divides, it would seem very unlikely that you get big shifts in the amount of the mutation between the daughter cells. And so in populations, though, and in families like this, we do see big shifts. So here, the mother has 11% mutant mitochondrial DNA in blood and urine, and she's had children where it's gone down a little bit or it's gone very high. So that's suggesting it's going through a bottleneck, that perhaps rather than all the thousands of mitochondrial DNAs being randomly sort of passed on to a daughter cell, at some stage it's gone through a genetic bottleneck where a small number of mitochondrial DNAs determine how many mutant mitochondrial DNAs there are in the oocyte. And so, this mutation appears to have arisen perhaps in one of the oocytes of the grandmother. The mother's had a small amount, and then it's amplified up in the next generation. So if we're going to think about options like prenatal diagnosis and pre-implantation genetic diagnosis, things like that for diagnosis or prevention of mitochondrial DNA mutations, what are the key issues? And one of them is when is this bottleneck occurring? And that was unclear for a long time. I think it's pretty clear now, or there's perhaps a little ambiguity around it. I'll show you some of the evidence. If you were thinking about prenatal diagnosis, so looking at chorionic villus samples or amniotic fluid in an established pregnancy, there's no real technical problem in measuring the amount of the mutant DNA in a sample. The question bedeviling this field, at least initially, was will what you measure in the chorionic villus, which is going to go on to become the placenta, reflect what is present in all the tissues of the fetus, and what you measure at 11 weeks of gestation, is that going to be the same at birth, and will it change afterwards? And then once you've got a number, a number's great, but how do you interpret that number? So it's very easy if it's 0 or 100, and for some mutations like the 8993, we've got a pretty good handle of exactly where a cutoff might be. For other mutations, that's much more ambiguous. So it's a real issue around how do we predict the clinical outcome from the mutant load. So that's for prenatal diagnosis. For prevention, clearly donor egg IVF is an approach that can allow families at risk of mitochondrial DNA mutation to avoid having an affected child, but there are pros and cons of that method. Pre-implantation genetic diagnosis can be very suitable for some families, but there are pros and cons. And so it's clear that many families with mitochondrial DNA mutations, those approaches will not be suitable for, and so that's why there's been an incentive to think about approaches like mitochondrial transfer or nuclear transfer to demonstrate whether they can be feasible, safe and effective. And I have to confess that 10 years ago I thought it was science fiction, and then some landmark papers in the macaque monkeys, the studies from Oregon, and the studies from Newcastle in human embryos made me think, wow, this is amazing. This is really something that could come in the course of a few years to be an appropriate approach. So I mentioned that when does the mitochondrial DNA bottleneck occur, and really Eric Schubridge's group presented some beautiful data in 1996 in mouse models showing that it happened very early in oogenesis, by the time of the primordial germ cells, well before the development of the mature egg. So that's important because if the bottlenecks occurred before the egg is released, then we don't have to worry so much about it changing afterwards. There's just a few little wrinkles on that, I guess. So does that apply in humans? And I guess the first study there was one that we published from Melbourne with the help of Deborah Gook, at the time at what was then the Royal Women's Hospital Reproductive Biology Unit, looking at a family we diagnosed who had 3 children with high amounts of this mitochondrial DNA mutation, the same one I mentioned before, 8993, theta G, 2 with Lee syndrome. One had been diagnosed as SIDS, but was clearly really a mitochondrial death rather than a sudden unexplained infant death. And we had the opportunity to look at 7 oocytes from that mother, and found this rather remarkable result that 6 of the 7 oocytes were almost completely mutant, and 1 had no detectable mitochondrial DNA. And the American Journal of Human Genetics made us go through quite a few hoops to prove that was real, and I think everyone now believes that this happens in the families. So that's implying that in human oocytes, these have clearly gone through this bottleneck, well before the oocytes have been released, have matured, and those marked distributions that we see in individual family members appears to have been present in the oocytes. So quickly through these sort of options, prenatal diagnosis by CVS or amniocytes has a lot of attractions in terms of cost compared to other IVF approaches, likelihood of obtaining a pregnancy and access, no need for donor gametes and so on. The real challenge with prenatal diagnosis is that once you're doing prenatal diagnosis, you're committed to a pregnancy. So the outcome is, if you predict it's going to be an affected pregnancy, to avoid an affected child requires termination, and that's something that many families are not comfortable with. And also, in some cases, the outcome is going to be black and white in terms of that prediction, and in others, it's not going to be. So if you do these sort of things, you have to accept that there could be some areas of grey in interpretation. So it depends very much on attitudes of families to pregnancy termination. And as a generalisation, it's really only suitable for women with quite a low likelihood of having another affected child. So as an example of that, this is that first family I put up before with an affected boy and 3 children, 3 women, one of whom had prenatal diagnosis performed at the Victorian Clinical Genetics Service, and no mutant mitochondrial DNA was found, and that's really consistent with the bottleneck that if you're starting with a small amount, it's more likely to go to 0 than to hang around there. The second sibling also had prenatal diagnosis, and the same result, no detectable mutant mitochondrial DNA. The problem with these sort of families, I guess, in terms of counselling, is that we had counselled these 2 women specifically about them being in a good situation for attempting prenatal diagnosis. Their sister had had no counselling and turned up pregnant expecting prenatal diagnosis, and our recommendations had been that a woman with that higher level of mutant mitochondrial DNA, prenatal diagnosis was not a great option because it was very likely to have an uncertain outcome. But once they've turned up in the clinic, there's really a responsibility to counsel them about the limitations and go ahead with the test if they choose it. So she went ahead with CBS, and that showed a mutant load of about 70%. What do you do with that number? Well, back in 1999, we'd done a pretty extensive study on all the patients with this mutation and came up with this logistic regression model where we could predict the likelihood of a severe outcome based on the amount of this mutation, and that's really the only mutation that these sort of data are available for. And so you plug in the numbers to this model, and it predicted that there was a 37% probability of this child being affected, of this pregnancy being affected, with 95% confidence intervals at 22 to 55%. Now that's clearly not a result anyone wants to receive or convey, and in this circumstance the pregnancy was terminated, and that's clearly illustrates the limitations of prenatal diagnosis for women at substantial recurrence risk. So pre-implantation genetic diagnosis, again, I'll go through pretty quickly. Mary's going to say much more about this, I think. This technique has been around for quite a while now, used widely for many single gene disorders. Again, has some attractions in that there's no need for donor eggs or pregnancy termination using PGD. And this was first done for mitochondrial DNA mutations, I believe, by Julie Stephan in Paris in 2006, and the Paris group have done most of the work in this area, although I'm aware that PGD has been done in Maastricht and Melbourne and Newcastle. I'm not sure of anywhere else. Realistically, you'd only want to implant embryos with very low, preferably 0 mutant loads, but they may or may not be available. And so, in other words, that technique can only be available if the oocytes that are harvested and the embryos that are generated have those low mutant loads. So if a woman is homoplasmic, has 100% mutant mitochondrial DNA, then PGD will be absolutely no use to attempt. And if she has a very high amount of mutant mitochondrial DNA, chances are that there may not be many eggs that would be suitable for implantation. One of the useful features of PGD, though, is that if a woman goes through one cycle, even if it doesn't result in an embryo being implanted and a successful outcome, it at least gives an indication of what is the distribution of mutant loads in her eggs. So I'll finish by showing one family here that we've done prenatal diagnosis and PGD on. Again, I use the term we rather liberally. So the proband here presented at 4 months of age with severe failure to thrive, lethargy, hypotonia, developmental delay, hyperlactic acidemia, progressive deterioration leading to death at 26 months of age. The parents were counselled after this particular mitochondrial DNA mutation at 8993T to G, again the most common mutation we see in children with mitochondrial disease, about options such as donor egg IVF, prenatal diagnosis, PGD. They elected to have prenatal diagnosis in the first pregnancy. Unfortunately, that had a very high mutant load of 95%, very clear prediction of an affected outcome. They then weren't going to have any more children, but came back to clinic when they were unexpectedly pregnant. Prenatal diagnosis performed again and again, a very high mutant load and an affected outcome. So, really not a great series of events. But in discussions with Sharon Stockmeyer, who's here today from Melbourne IVF, and Leander Wilton, we thought this was a family well suited to offering prenatal pre-implantation genetic diagnosis. And this is an outline from the first cycle. There were 7 embryos obtained, 4 of them here with almost 100% mutant loads, 3 with somewhat lower loads of 30–40%. And there were also a number of unfertilised eggs, one of which had quite a low load and the others all very high. The embryo with the lowest mutant load was implanted. pregnancy was established in the sense of obtaining a positive beta-HCG, but unfortunately it was a blighted ovum, and so that was not a real pregnancy. The family went through a second cycle of ovarian hyperstimulation. Only 3 embryos obtained in this case. Fortunately, one with a very low mutant load of 2.5%, and a couple of embryos with very high loads, unfertilised eggs with very high loads. This was implanted. CVS recommended to check what the mutant load was going to be, but you can understand this was a very precious pregnancy, and the parents opted not to have CVS. So, healthy child was born. Mutant loads of 2.5 to 4.5% found in cord blood and placenta. So, a very good outcome for that family. So I've tried to quickly cover, Oxford disorders occur in about 1 in 5,000 people, any symptom, any organ, any age of onset. Currently we know of about 215 disease genes. Nearly all of the mitochondrial DNA genes have mutations causing disease. Treatment is mainly symptomatic and limited, putting the impetus on being able to offer families prevention. CVS analysis has a place, so prenatal diagnosis, but best for women with relatively low recurrence risk. PGD avoids the need for donor eggs or pregnancy termination. One cycle can provide valuable information, but it does have these limitations that, all of these approaches have some uncertainty around them, and that's not really unique to mitochondrial DNA disorders. I think people get a bit precious about uncertainty. Uncertainty is a day-to-day thing with genetics, and so long as families understand that, it's not a reason to not offer things, provided you believe they're safe and the families understand the issues. Basically, any of these reproductive options will only suit some couples, and as I've said, prenatal diagnosis, PGD, are not suitable for women with a relatively high recurrence risk, and so hence the desire for approaches like mitochondrial transfer. I just thought I should mention that the prenatal diagnoses I've talked about were done in the VCGS lab associated with Murdoch Children's, where the mitochondrial section is run by Belinda Chong, and the PGD I talked about was done by Sharon Stockmeyer and Leander Wilton. And I'll leave you with some images from the bloody long walk of 2014, 15 and 15 with the Murdoch mitochondriacs there. Thanks very much.
Off the walk.
Bloody good.
Yeah, bloody good.
Apart from the blisters.
So, is there any questions for David? Yeah.
Actually, the topic is very good. I wonder for the mutant load for the mitochondrial DNA and ectoplasmic disease, does that actually vary in different cell types in the human body?
Yeah, so that's an interesting question. So for the particular mutation that I talked mostly about, the 8993 T to G mutation, it seems that what you measure in muscle, blood, urine, brain, or post-mortem samples seems pretty much the same. Clearly, oocytes have very different levels. Intriguingly, when you look at blood, the average mutant load is the same as other tissues, but if you look at individual lymphocytes, they can have quite wide mutant loads. So that mutation, it's partly why I described it as well-behaved, that every organ, the average seems to be the same. For many other mutations, for the ones we see, for point mutations, almost all the data suggests there's very little segregation during embryogenesis. So at birth, the mutant loads appear to be pretty much the same, but then some mutations are selected out of blood, for example, and can go up and down in other tissues. So it depends on the mutation, and there's definitely the prospect that what you measure in one tissue may not reflect what's present in another.
And for the clinical diagnosis, it's usually blood cell.
So it depends. So for mitochondrial DNA, in a child, it's very rare to lose mitochondrial DNA mutations quickly. So blood is usually okay in children. Certainly in adults, blood is often suboptimal, particularly for mitochondrial DNA deletions and for some mutations like the MELAS point mutation. And it turns out that urine sediment is a much better non-invasive tissue. So urine sediment retains mitochondrial DNA mutations better than blood in terms of non-invasive samples.
David, the person with the 59% mutant load, are they more liable to develop something later in life? If they have a viral infection or if they smoke pot, are they likely to go off the edge?
Don't think I have the expertise to comment on pot. So It's hard to say an absolute no, but for that mutation, what we did was to pull all the data out of the literature, and there's always ascertainment bias in the literature, often not looking at the very oldest individuals, so it's possible they could still develop symptoms. Certainly, it's sister mutation, a T to C mutation at the same nucleotide. You have families where women can be asymptomatic with 95% mutant load in their 30s and 40s and develop symptoms later on. So it's very hard to give an absolute yes or no answer to that. For that particular mutation, all the data suggests there's a pretty low chance that she would develop symptoms, but it's not zero.
Thanks, David. That was a really clear talk. However, I've still got some questions, probably largely due to my own ignorance. So one thing I was going to ask is it trying to predict future health? And so people are presenting for testing, say a young woman who wants to find out and then is trying to think about whether she might consider having children and what her risk might be. I suppose there's two elements there. One would be what interventions might be available to try and ensure that she can have a child free of mitochondrial disease. So for and some of the things you've mentioned around blood and urine. I'm going in and out here, sorry.
Yeah, it is a bit. Yeah.
With blood and urine, do you suggest testing again at a later point, or do you mutation load change over time as well?
Yeah, it's hard to give the yes/no answers again, but as a generalisation, we regard urine as a much better sample for making inferences, particularly for things like the common MELAS mutation and deletions, where what you measure in an adult in blood is very likely to be an underestimate of what's present in other tissues. So urine loads, most of the data suggests that when you look at the load in urine in a 30, 40-year-old, it extrapolates back to what was present in the Guthrie card at birth, for example. So it seems like the urine loads are pretty stable, but there's limited data. There have been hundreds of these sort of samples tested, but it's not an absolute. Some mutations clearly can increase in different tissues, and particularly within tissues. So the classic findings in mitochondrial disease originally were the ragged red fibres and COX-negative fibres, which are reflecting individual muscle fibres, some of which have high mutant loads and some have low mutant loads. And yes, some deletions, again, it's very hard to do serial studies on tissues in individuals, but many of the data suggest that deletions might increase in amount in mutant load with age, where there's a replicative advantage for a smaller molecule. For point mutations, probably less clear, but some suggestion that the common MELAS point mutation might do that in muscle.
Right, there's no further questions. Thanks very much.